everyone, my name is Reagan Breme, and today for my interactive case study 7 on 5FU, I am going to be discussing this and how it seems to behave like a regular nucleotide until it doesn't. So it enters cancer cells undetected and fully disables their RNA and DNA production. So it's like giving someone a recipe book, but there's a hidden ingredient and you don't tell them what it is. So it's initially a great dish, but then halfway through, it's not the same as it should be. So I'm going to be explaining how 5FU is digested, how it affects the RNA production, and the chemical processes that make it a potent cell inhibitor. So first, why do scientists produce 5FU? My first slide is going to be touching this, and understanding the purpose clarifies everything. So let's proceed to slide 2.
Researchers discovered that cancer cells drew in uracils quicker than normal cells in the 1950s, and this finding was very important. This finding inspired researchers to create, fake uracil cancer cells that would mistakenly take in 5FU that was made that same way. So my slide here demonstrates how 5FU directly enters tumor cell pathways. It becomes poisonous to metabolites instead of benefiting the cell. So by metabolizing the medication, cancer cells, they're contained in themselves. So the important note before discussing RNA and DNA is that 5FU exploits cancer cells' uracil addiction. So after learning the why, let's examine 5FU's effects within the cell. Going on to slide 3, it is basically a detail on the metabolic journey of 5FU. A cancer cell handles 5FU as uracil once it enters. It modifies 5FU in the pyrimidine metabolic pathway, as my graphic illustrates these branches. RNA contains FUTP from one pathway. It creates FDUTP, which inserts DNA and causes replication mistakes. This branch produces FDUMP, which completely inhibits thiamidylate synthase. So DHFU to FUPA to FBAL is a secondary route that breaks down 5FU. It doesn't necessarily destroy cancer cells, but it explains why individuals react differently to the same amount. The key takeaway from this slide is that 5FU transforms into several chemicals that target various cell sections. So after mapping out how these metabolites are created, let's look into RNA, which is the first big target. Moving on to my next slide, which explains how FUTP hinders the RNA synthesis. So RNA damage is one of 5FU's first and most underestimated effects. Our metabolite FUTP nearly looks like UTP. Since RNA polymerase can't distinguish them apart, it inserts FUTP into developing RNA change. So my diagram depicts the direct diversion between FUTP feeding into rRNA synthesis and mRNA synthesis. So once FUTP is added, RNA fold strands improperly and the misassembled rRNA ribosomes break. The translation of FUTP containing mRNAs is unreliable because of this. Cancer cells depend on quick protein synthesis, so this is hard for them. It's like changing a plan halfway through something. RNA disruption is only one factor though. So we will analyze 5FU's most renowned target, which is the thiamidylate synthase. This slide focuses on the mechanism of thiamidylate synthase inhibition. So the 5FU's main molecular showstopper for anti-cancer action is thiamidylate synthase, which makes DTMP for DNA replication, and it attaches to FDUMP. And not only does FDUMP bind only for a second, it freezes the enzyme in place by forming a type 3-part complex with this folate cofactor. The cell cannot synthesize thymidine without thiamidylate synthase. Without thymidine, cells DNA cannot be created, and cancer cells that divide without adequate thymidine strike a wall called thymidinous death. This single inhibited step makes 5-FU so powerful against quickly proliferating malignancies. Since 5-FU affects RNA and DNA, let's now examine why cancer cells are more damaged than normal ones. This next slide shows that cancer cells are more chaotic and overloaded, while normal cells are more orderly and controlled. So cancer cells divide rapidly. They continually make RNA, copy DNA, and cycle through cell division checkpoints. Because of this, they absorb uracil and 5-FU quicker than healthy cells. FUTP or FDUTP in their RNA and DNA prevent cancer cells from repairing themselves. They keep going, and this forces them to fall apart. So slow, healthy cells may repair themselves, but cancer cells cannot. And metabolic strain renders them sensitive to 5-FU. So the combination of 5-FU attacks to its true potency. For our next slide, it is going to assemble everything. 5-FU's lack of specific focus of attack distinguishes it from early chemotherapy medicines. So my image to the right shows how cancer cells resist or react to this treatment. Many malignancies react effectively to 5-FU's multi-hit technique. So within the cell, FUTP destabilizes RNA, FDUTP disrupts DNA replication, and thymidylate synthase is inhibited by FDUNP. 5-FU overwhelms a cancer cell's capacity to adapt, mend, or escape by targeting RNA, DNA, and is a key enzyme. It is the key medicine to colorectal and digestive tract cancer therapy because its three-pronged pressure drives cancer cells toward cell death, which is apoptosis, rather than resistance. And now I'll conclude to my final slide. In conclusion, 5-FU or 5-fluorouracil acts by impersonating uracil, which cancer cells need. It becomes three strong metabolites that assault the cell on numerous fronts after absorption. Metabolize into FUTP, FDUTP, and FDUNP, each with specific targets. FUTP disrupts early RNA synthesis, FDUTP corrupts DNA replication, FDUNP inhibits thymidylate synthase and thymidine biosynthesis. Using many hits overwhelms cancer cells quicker, and they can mend, causing apoptosis, which is cell death. 5-FU is one of the earliest chemo medications. It is still used to treat colon, breast, stomach, and digestive system malignancies due to its simplicity and efficacy. Thanks for listening, and have a great day.